Hello and a very warm welcome to all of you on the fourth day of the 11th edition of Tata Literature Live, Mumbai's International Literature Festival. A huge thanks to our title sponsor Tata as well as our co-sponsors Tata Steel and Tata Projects. This session is presented by the Blank Slate Knowledge Foundation, Mumbai. Welcome to our session this morning as part of our 360 at Campus Festival, which is a small part of the larger Tata Literature Live, Circuits of Sensation, where we explore the development of the sensory pathway, the circuitry that controls how we experience the world. And by the end of the session today, you may wonder whether there is a boundary between reality and illusion. Taking us through this exciting journey today is our speaker, Dr. Shubha Tole. Dr. Tole did her doctorate from Caltech, USA, and was a postdoc at the University of Chicago. And she then joined Tata Institute of Fundamental Research in Mumbai, India, as well as a faculty member in 1999. She has received many awards for her research, as well as being a fellow at all three Indian scientific academies, which is INSA, IAS, and NAS. Now remember, you are just an important part of this session, as we all are, and we would love to hear from you through your questions that you can put in on your chat boxes or your comment panels on whichever platform you're watching. So do remember to keep shooting us your questions along with your names so that we have a whole bunch of things to ask Dr. Tole once she's done sharing her views and her thoughts on the circuits of sensation. So let's kick off today morning's session, the circuits of sensation, bringing the world to your brain. Over to you, Dr. Tole.
Thank you so very much. It is a delight, a privilege, and it's something I'm really looking forward to share the excitement that we neuroscientists experience every day. Because we study this amazing computer in our heads that gives us the ability to question, to think, to look, to enjoy everything. In some sense, we study the very circuitry that gives us our lives, that gives us our world, that gives us our experience of it. And today I would love to share some of the excitement of this research with you. I'm going to talk about why brain circuitry is important. And then I'm going to showcase small bits of findings from my lab, from my students and postdocs, that we've contributed to the vast landscape of neuroscience and the vast landscape of everything that we know about the brain. And then I will look forward to conversing with you folks. Any questions you may have. The questions are the most important part of the session. So please keep them ready and keep them coming. And I look forward to this. So I'm going to share screen now. This marvelous image. This is a view of all of the wires in our brains. This scary, riotous mess of complex, intricate circuitry. This is what makes us who we are. This is what gives us our perception of the world. This is what brings the outside world into our brains and in some sense creates this sensation of the world we exist in. This is human brain circuitry. This is not a model system. This is not something we can experiment upon. But we ask how such circuitry is created across evolution. In model systems, in zebrafish, in Drosophila, the fruit fly, in mouse. Because the mechanisms that made this beautiful circuitry are the same, whether you look at a tiny little winged insect or whether you look at a fish or a mouse or us. The evolution has conserved how this marvelous computer is formed in our heads. Why is circuitry important? Why are the circuits of sensation so central to our experience? I'm going to walk you through some experiments, as you will. We're scientists, so we do experiments. Nothing bad will happen, I promise you. I'm going to walk you through some little tests to bring out how completely dependent we are on our circuitry for what we perceive. Am I still sharing screen? I think something's... There we go. Okay. It's well said that seeing is believing, and I'm going to show you a few flowers. I think everybody would agree with me that these are, beautiful flowers, and I think most people would agree that these appear to be different flowers. But you know what? These are the same flowers, just photographed using a different lighting, using ultraviolet light. Okay, so we see, since we don't perceive ultraviolet light, we see the flower as purple and white with a yellow middle, and here we see a pretty yellow flower. But if one could see in ultraviolet light, okay, as insects can perceive, one would see that these flowers have patterns that are unseen by us. Okay, now since we cannot see ultraviolet light, this coloring is a false coloring to show us that there is a central patch and a surrounding patch, and the two are different. Here there's a central dark patch and a surrounding patch, and you can imagine that for insects for whom it is important to be able to find the center of the flower, this kind of bullseye pattern is perhaps more useful, right? So if one can see in a type of light that humans don't perceive in, the flower looks completely different. Now, what color is the flower? That becomes a philosophical question, right? Because it depends on who's looking. So not only is our wiring, the properties of our neurons, not only do they give us the perceptive color, but they also limit us. Because if they cannot perceive ultraviolet or infrared, they're not giving us part of the world that's out there. So in fact, neuroscience is at the same time fascinating and humbling, because it tells us how much our circuits work to give us the sensation of the world, but it also tells us how limited we really are and how puny and, constrained we are. It's sort of like we're living in the matrix where the information that's funneled to our brains is the information we have, and we think, oh, we're powerful humans, we change the world, we build buildings, we this and we that. Well, we only see what our sensory nerves can give us. So that's why studying these sensory nerves becomes very important, and acknowledging that the limitations of our circuitry are our limitations. And there's a whole world out there that we have no idea of. Okay, I'll test this a little bit further. I think you would all agree with me that, there is an orangey rectangle and a browny rectangle, and that there is a brownish dot in the middle of the orange rectangle, and there's an orangish dot in the middle of the brown rectangle. Well, our circuits have trapped us again. The properties of our neurons have trapped us again. Context is everything. We perceive differently based on context. Okay, I've just copied this brown dot, and I'm just going to recopy the square again and again and again. It's the same square. I'm not playing a trick on you. And now maybe you will agree the two colors are perhaps more similar than you thought. In fact, they're the same color. Okay, it's the same color. And yet my eyes, and perhaps your eyes, will still insist that this is browner, and then this thing sort of transits from brown to orange, and this is orangier. We can't help it. Okay, the properties of our nerve cells are like this. So context is everything, and context colors what we see, literally and metaphorically. So in terms of knowing what the world there is, our circuits are what we have, and they're limited and they're constrained. I'm going to now show you an experiment that in fact tests whether you hear with your ears or your eyes. Okay, this is a well-known experiment in psychology called the mcgurk effect. Here we go. Tech team, please let me know if you cannot hear the sound because this has video and audio.
Ba, 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 ba.
Okay. I mean, yeah, baby sounds, ba, ba, ba. There was no test there, no challenge. I'm sure everybody heard that. And now let's see the next one.
Ba, 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 ba.
So, I suppose many of us heard fa, fa or va, va, right? Well, it's going to knock your socks off when I tell you that it was the same soundtrack playing both times. Okay, it was the same soundtrack. And to prove it to you, here now is that same soundtrack and both the videos on one screen. You can look at the one and you can look at the other and you can watch the percept of the sound change in your heads. In fact, if you close your eyes, you'll hear the soundtrack as it was recorded, and I won't tell you which one it was. And then when you open your eyes, depending on which face you look at, the percept will change.
Ba, 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 ba.
Amazing, right? It works every time on me, even though I've seen this clip a million times. It works on me, even though I know what it's supposed to be. I know that the soundtrack is supposed to be ba, ba, ba. But every time I look at the one face, I hear ba, ba, ba. And when I look at the other face, I hear ba, ba, ba. I cannot help it. Okay, I cannot help it because my brain is wired this way. And neither can you. Okay, so this is really how our percept of sound is colored by what we see, right? You've seen this at parties, right? You've seen this in crowded rooms where you're trying to listen to a conversation, maybe somebody far away, and if you look at them and if you can see their lips move, you can somehow hear them better. So that's what it is. We're using the mcgurk effect, right? So it really does seem that we hear with our eyes, huh? A little bit. Okay, let's move to the next sensation.
Ba, ba, ba.
Touch. Touch. I'm going to now ask you to do a a mental experiment, okay? I'm going to ask you to imagine a tennis net. We've seen one. If you've never seen one, here it is. And now I'm going to ask you to close your eyes and use this wonderful probe that biology has given us, our fingers, okay? You're going to close your eyes and you're going to use this probe to test whether this material has holes in it. Okay, close your eyes and imagine the tennis net and you're feeling it and you're poking your fingers through it and you have to answer the simple question, does this material have holes? Yes, no? I mean, not only can your fingers go through it, the whole hand can go through it. Okay, now close your eyes and do the same experiment with a mosquito net. Okay, you're feeling it with your sensitive probes and you're asking, does this material have holes in it? Okay. I don't think anybody would be able to discern if the mosquito net had holes in it. At the most, you would feel the surface to be a bit rough, but you can't discern it has holes in it. And why can't you discern it has holes in it? You can see the holes with your eyes, right? The problem is resolution, okay? We're using this great big probe of our big fat fingers, fat with reference to the mosquito net, okay? If I now gave you a toothpick to hold and asked, does the mosquito net have holes in it? You'd be able to tell with a toothpick, right? So this is obvious to you. Why am I belaboring the point? My point is that based on the resolution of the probe, okay, and based on the processing computer that processes the information, we see different levels of resolution in the world, okay? If we use our eyes, we would see that the mosquito net has holes. If we use our fingers, touch, we would not know that it has holes. If our whole bodies were the size of an ant, okay, when an ant, you've seen these lovely cartoons, right, where the world of an ant is depicted. If an ant were to walk on a carpet, the ant might think that it's walking on really, terrain with dense forest in it, right? Whereas we walk on the carpet and we see it as a flat surface. So we have built our world to the scale that we need. Okay, we built our world with chair, table to our size. And we build it with fine mosquito nets to keep the insects out because of their size. And we think this is the way the world is. But if we could see at a micro resolution, okay, such as we do with microscopes, for example, there is a whole world inside every single cell. There is a whole world inside every single atom. And we don't know it's there because our senses are limited unless we expand our senses using these gizmos. Right? So what our brain thinks of as the world is at once rich and diverse and wonderful, but also constrained and limited. Okay, so the constraints of our circuitry are the constraints that we live with, unless we acknowledge, know, and then choose to cross them. Okay, so this then is why studying circuitry becomes terribly important, and this is why studying how the circuitry is formed in our brains when we are but embryos, that becomes important. My work, my research work, and that of my lab, the team of wonderful students and postdocs with whom I've worked at the TIFR, the Tata Institute for Fundamental Research, for 21 years now, we delight in exploring how to build a brain, how the brain is built, how this marvelous computer is assembled. Okay, so you can't go and buy parts. Okay, you can't buy motherboards and things and sort of figure out how to wire it all together. You have to build it. And would that we could build it, you know, with a team of technicians coming in and look at this one planting in a bright idea, haha. Every part has to be grown. Every part has to be fabricated as the embryo develops. And then the wires have to be connected, and they have to be connected just so, in order that the little human being that appears in the world can function, can perceive the world, and can make sense of it, and then can take action. So here are two videos that bring out this process of how an embryo is built, because within the embryo will grow the brain. On the one side, this cartoon is a human embryo, and on the other side, this is a fish embryo. Okay, and I want to sort of bring out the parallels to make the point that evolution has really preserved the same basic mechanisms in growing complex organisms from very simple structures. So here is one cell, and it's soon going to become two. And here is one cell, and it's soon going to become 2 and 4 and 8 and 16, not waiting for us. And the marvelous glory that is development is playing out. Here, the ball of cells has invaded the placenta, and the human baby is growing there. This blue line is what's going to actually form the embryo. And you can see that that blue line now, I've paused this, you can see the beginnings of the head. There's a tiny eye there. This is the beginning of the hand popping out. And you can see this little dotted structure. This is the beginnings of what will be the vertebral column, the bones of the spine and their musculature and so on. And meanwhile, the fish has done the same thing. Okay, you can see the fish too has now extended a very long tail, and you can see its spine and bones and so on forming. Let's let the human catch up. And that's the massive yolk, and that's the enormous eye of the fish. And here's the human growing, and you can see the little hand coming up, and now the leg, and now the eye is more like an eye. And here on the fish side, the little heart beating. Here's the umbilical cord. Ears formed, all the sense organs coming in place. Both embryos building themselves. The cells growing. The structures forming. And here the fish is saying, sayonara. Okay. So this gives me great joy to see. This magic and marvel of development gives me great joy. How much complex architecture has to form. And it happens beautifully and in synchrony. And every little animal that's born brings this about. Every egg, every little mouse on the street, every cat, every insect, and every human pulls off this marvel of development. Okay? Now for the human to reach this stage takes 9 months. And at the end of it, the human can't walk. The fish, in 85 hours, in 3.5 days, has disappeared from our screen, right? What's the difference? Well, the fish has its nutrients packaged in an egg already. For the human, it has to build the whole placenta and so on inside the mother's womb, and that's why it takes longer. But also, the human invests a lot of time in creating the basic architecture for the brain. Much of the human embryo's development is actually to set up the brain that will continue to develop after the baby is born. And that is why a human baby can't start walking right away, because the action is still happening here. That's a broad sweep of development, but let's look at what's happening in the brain itself. Okay, this is a small video of a section of a mouse brain. This is a slice of a mouse brain in which there are some red labeled newborn neurons that are going to be zipping around, going to their destinations. This was published research from a colleague, Kazunori Nakajima, from Japan. And here, look at these beautiful neurons in the embryo mouse slice. Look at them gorgeously zip around, extending their processes. Okay, so each cell, although they're all doing somewhat different things, each cell knows where it's going. Okay, each cell has a destination. Each one is listening to cues. This is the type of work that developmental neuroscientists do, to ask how is the hardware created? Okay, how are the neurons created, and then how are they wired up? Okay, once the neurons are in place, they have to form circuitry. Okay, here's a brain cartoon, and here is a simplified version of all of the circuitry that we need to see. It's a heavily simplified version. I'm not going to walk you through it. It looks like a complex train map, right, for a big city. Well, it gets even more complex. Here is one neuron. Here's what one neuron looks like, okay? And each of these wires of the neuron either receives signals from other neurons, or one of them sends out signals to the next neuron. Now, the human brain has 100 billion neurons. Okay, and each of these 100 billion neurons makes between 1 and 10,000 connections with some other neuron. This is the complexity of the circuitry we're talking about, and this is why this picture is at once amazing and fascinating and terrifying. Imagine trying to study this, right? So we study the sensory pathway and how it forms and how the circuitry is wired up precisely in the mouse, where it looks simpler. So here this is a slice of a mouse brain. And in green is the sensory nerves coming in to the cerebral cortex. This blue structure here is the cerebral cortex, and you can see this green bundle of wires as they've grown, okay, to the cerebral cortex, bringing in sensation, bringing in vision and hearing and touch to the mouse. And of course, these senses are as important to the mouse as they are to us. And having them at high resolution becomes even more important. Why high resolution? Now, if you have a limited amount of processor, okay, to process the signals that are coming in, it would be very expedient to devote a large amount of that processor to the signals that are important, right? You would want to process at high density and high resolution important signals, and perhaps at lowish resolution signals that are not as important. So what is important to us, okay, when humans experience the world by touch? Okay, you've seen babies do it, we do it, we use our fingers, we use our hands. Babies will sample everything with their mouths. Okay, our lips and our tongue, the touch is very important there, because you have to be able to feel the texture of the food when you eat it. Right? So there are certain parts of our body where we have high resolution of touch. How does the brain give us that? Very cleverly, the brain divides much more of its processing territory to areas from which it wants high resolution sensation. So here is a picture of how we would look, okay, if our bodies were scaled to the area of the brain that is devoted to processing sensation there. Right? So this is telling you that our mouth, our tongue, okay, to a good extent the rest of our face, and certainly our hands have large territory in the brain devoted to processing touch, and that's why you can get a high-res sensation. High resolution means that if you take two pin pricks and at two different points, you'll be able to tell they are apart even when they are very, very close on your hand. Okay, but if you do the same thing out here, you won't be able to tell that two are two. You will think of them as one just because you don't have the resolution. Right? Okay, the mouse does this too. The mouse does this too. And for the mouse, The same regions are important, the lips and jaw, the forelimbs, but for the mouse has one more appendage that we don't. The mouse has whiskers. And the mouse's whiskers actually whisk and sense the environment. Before a mouse goes through a gap, it will actually whisk and sense whether the gap is big enough for it to go through. Okay, mice do this, cats do this. Yeah. So here is our mouse. And here are where the whiskers would connect in the front of its nose. And here is how the mouse would look if you drew it based on how much territory in its brain is devoted to each body part. So you can see, much like us, okay, the face, nose, jaws, upper, lower jaw, and certainly the forelimbs have a lot of territory. And look at this. Each little whisker pad has so much brain territory and not so much the rest of the body. Okay. So this is how the mouse and its sensory apparatus that senses the whisker pads become a way we can ask how sensory nerves grow into the brain. Now, here is a cartoon on one side and the same mouse slice on the other side. And this is the PhD thesis work of my former student Suranjana Pal. She asked, well, how does this red nerve bundle enter the cortex? How does it know, here now in green on this side, how does it know to enter the cortex, and what are the things that control it? So Suranjana tested the role of a particular gene that we hypothesized would be important in this process. And for this, we used something called a gene knockout mouse strategy. Now, what's a gene knockout mouse strategy? Okay, for people who've never heard the word, all the cells in our body have all the genes, right? They have a copy of every single gene, but each cell uses information from only genes that are important for that cell. For example, a blood cell will use the gene for hemoglobin to make hemoglobin protein, and so on. Now, you can knock out a particular gene to ask, well, how do those cells behave without that gene? And let me give you an analogy. Suppose you've never seen a clock before, okay? I think much of this younger generation has only seen digital cell phones or something. So an old style clock with all the gears and so on. Suppose a clock just dropped in from the sky, okay? And you noticed it had an hour hand, and it had a minute hand, and it had a second hand that went round and round. And you said, okay, these gears move and those hands move. I hypothesize that these gears have something to do with the movement of the hands. And I'm going to knock out this one gear and ask how the clock behaves without it. So you knock out that gear. And you notice that the hour hand is still moving, but the minute hand is stuck. And then you say, okay, this gear may control how the minute hand moves. Now, you have to be careful with this because a knockout strategy, you've just removed that gear. Note that the movement of the second hand may also be stuck. And the movement of the second hand may not be directly controlled by the gear you knocked out. It may be related to whether the minute hand has moved or not. Right? So your effect of your gear may not be direct. It may also be indirect. So these are the kinds of analyses that you have to do when you do a gene knockout and you examine the brain. And this is why a PhD takes 5 years. Okay, because you have to do careful experiments and ask precise questions. And I'm going to show you one slide of Suranjana's data. Okay? She asked, how does the circuitry of sensation form in the brain? How does this nerve tract get into the brain? And here we have the mouse. And here we have an image of a slice of a normal mouse brain. You can see that green bundle that I showed you, that was in the embryo. By the time the mouse reaches adulthood, you can see how the green bundle has made these little clumps in the cortex. Okay? And here it is at high mag. You can see these clumps. And each thin little green line is a nerve. And each little clump actually brings in sensation from one whisker. Okay, literally, the sensation of one whisker connects to one of these clumps. So now we have a way of seeing, well, how is this circuitry of sensation wired up in the normal mouse? Then what Suranjana did is she looked at the mouse in which this particular gene that we'd hypothesized was knocked out from the cortex. Not from the nerves growing in. The nerves growing in were just fine. But the gene was removed only from the cortex, the territory that the nerves are going to enter, and see what happens. In this gene knockout mouse brain, it's the green nerves enter all the way up to here, but they don't penetrate. Okay, for some reason, they do not enter the thickness of the cortex and they are unable to make these whisker-specific connections. The same thing applies for the visual system as well. These sensory nerves enter under the cortex, but they are not able to make connections, which means that although nothing is wrong with the mouse's eyes or ears or skin, and nothing is wrong from the nerves that took sensation from these organs to the brain, because we didn't knock the gene out in all these nerves, just because we removed the gene from the cortex, this mouse can neither see, nor hear, nor feel touch, because the nerves have not made the connection to the central processing unit that is the cortex. So now the next step in this project, okay, science is not something that is packed and okay, all done and solved. It's the next step. The next step in this project is to ask, well, what does this gene do in the cortical cells? What does this gene do that perfectly good sensory nerves, when they come in, are somehow not able to do what they're supposed to do? Okay, what does this gene control? And to do this, we are taking multiple directions of work. So I wanted to give you a little sense of the excitement of how research is, right? Each answer leads to the next question, and each answer leads to the next question. And now I'm going to showcase one vignette from an equally cool finding, this time in the structure that handles learning and memory in the hippocampus. Not the hippopotamus, but the hippocampus. Now, our senses and our experience of the world are all very well, but if you cannot remember what you have experienced, okay, you have a very weird life, right? In fact, it's safe to say that who we are is a collection of all our memories and all our experiences. Otherwise, we would just be naive like the day we appear in the world. So, we're also studying this structure that handles how memories are recorded, okay? How to build a recorder for memories. I could do a 14-part how to, you know, tame the brain, steal the brain's jewel, the dragon series. we could have a whole brain series on this. So how to build a recorder for memories. Now, the hippocampus does critical functions in taking in new memories, okay, and taking in new experiences. And I'm going to show you a cartoon of how the hippocampus's neurons change with experience. So here is a cartoon of a standard mouse cage. There are two mice. There's water, there's food, they're living their lives. And here is an enriched cage. What is an enriched cage? This environment has toys. Okay, there's a little wheel to run, there are tubes to crawl through, little building blocks. Okay, you can almost imagine mice like children in a playground, sort of exploring everything. Or children like mice exploring things. Now, if mice are compared, if their brains are compared, if the neurons of the hippocampus are compared, after living in a standard cage versus an enriched cage, okay, the neurons in the standard cage mouse are like this. There is a cell body, there are input wires that bring in signals, there are output wire. And here are the neurons of the mouse in the enriched cage. Okay, the hippocampus has more neurons, but not only that, each neuron is more complex, it has more wires. So experience of the world changes the brain circuitry. It changes how the neurons behave, it changes the number of their connections. Okay, we found something very cool about how brain stem cells make these neurons in the hippocampus, in the recorder of memories. Okay. We found a gene that has a critical function in these stem cells, and when this gene is on, okay, when it's being expressed, when protein is being made, the stem cells produce neurons. And when the gene is off, the stem cells produce an important kind of support cell, okay? Not a neuron, but the support cell that supports it. So it's the same stem cells that produce both the circuitry and the support structure. Okay, this work in one slide is the summary of the PhD theses of two marvelous students of mine, Lakshmi Subramanian and Anandita Sarkar. Okay, what they found was that if you prevent this brain stem cell from turning the gene off, if you continuously have it on, then this cell only produces neurons, and then you have more neurons in the hippocampus. Okay, so this is a switch, right? This gene acts like a switch where it controls how many neurons and how many support cells are produced. And remember, you need each, so it may not be necessarily a good thing to have only neurons in your brain and no support cells. And this is something that a postdoc in my lab, Archana Iyer, Dr. Archana Iyer, is looking at, and I'm going to show you some of her work. So this is a normal mouse brain. And here is a mouse brain with extra neurons in it. Each green dot and each beautiful green arbor. So here is a high mag, okay, of this gorgeous image that Archana took. Okay, so what Archana is asking is, this mouse with extra neurons, do these neurons wire up correctly? Are they helpful? Do they enhance the mouse's abilities? Or do they in fact interfere with the functioning of the perfectly good other, normal neurons, right? Having too many neurons may or may not be a good thing. We have to keep our minds open to it. So Archana is actually doing experiments to ask, well, are these extra neurons actually good for the mouse? Are they as normal? Are they like the normal neurons? So these are the kinds of questions that we're asking in the space of how our memory, how is the structure that records our memories made? Okay. Well, I've shared with you 2 vignettes of research, okay, about this marvelous structure, the brain. We don't work on the human brain. We use the mouse brain as an excellent model to study circuitry. How is circuitry formed? I opened with bringing out why circuitry is everything. Our brain circuitry give us our sensory perception. They allow us to perceive a range of things. They also constrain us, and they interact in interesting ways where we're sometimes using our eyes to color our perception of sound, and they allow us resolution. Okay, how our brains devote more area to sensations that we want to have better resolution in, and how this system is alive. How, when we experience new environments, when we record new memories, our brain cells grow, they sprout more in enriched environments, and how the system is controlled. How a stem cell has to decide whether to produce a neuron or a support cell. And all of these mechanisms together come together to create this magical, marvelous, fascinating structure. That is our brain. That allows us to be who we are, to grow our abilities, our personalities, to experience the world and to interact with it. Thank you very much. It's been a delight. I will now look forward to taking questions, as many as you wish, and I will end the sharing of the screen and wait for you folks to start a conversation.
That was a fantastic presentation, ma'am. I must tell you, in school, I was never really interested in bio, but I was riveted this morning. It was really so interesting, and you made it so engaging with your exuberance for the subject. It was so palpable.
Thank you so much. I always say, so I hope I'm speaking with students who are not necessarily doing science or not necessarily doing biology. I hope I'm speaking with an audience that is, each one of whom is pursuing their passions, right?
Right.
I mean, no, everybody doesn't have to study the brain, but whatever you're doing, please study it with as much joy and as much passion. One life to live, right? Let's take it as far as we can.
Absolutely. So we have a whole bunch of questions for you. Let's try and see how many we can pack in. Our very first question is from Shyam Sundar. He asks, are all our perceptions common? That is, can we generalize all experiences of one species as to be common, as exactly the same? Or does each human's perception change?
Okay, this is directly into the realm of philosophy. It's really early in the morning for okay. It is impossible to prove that the red I see is the red you see. Okay, it is impossible because your perception is your perception. It is what your circuits have given you in your brain. And realizing this is, I think, the first step for, human conversation, to recognize that people have their percepts in their head and they are as valid. So this opens a huge different conversation about open-mindedness and tolerance, and people perceive what they perceive, and we use our very limited ability of language to try and connect. Would that we had data cables, right, from one brain to the other. But language, prose, poetry, literature has all evolved so that we can communicate with each other and try and understand each other, because it's impossible to generalize.
Right. Our next question is, some people dream in color while some people don't. So what is exactly the truth?
Many of my answers are going to be, I don't know. Okay, one of the first things a scientist learns is that the sooner you learn how little you know, the better a scientist you're going to be. Our dreams are shaped by myriad factors, okay? First, what you remember. Many of us don't remember the details of our dreams. Our dreams will be affected by whatever else is going on in our lives. On Sunday, at 11 o'clock, I'm going to be conversing with a fantastic neuroscientist, David Eagleman, who has a beautiful theory about why we dream. So, and he's written about this in this book, so I encourage you to tune in there. Nobody knows exactly why we dream. I read some literature done in songbirds who, much like human language, they listen to sounds when they are little, and then they go through a period of crystallization where the sound is sort of shaped in their brains, but they're not speaking yet, they're not singing. And then when they sing, they rehearse and they improve their presentation without hearing any new input from their tutor, from their parent. They compare this with a recording of this song that they heard when they were little, and they improve. Now, a group of scientists recorded from songbird brains when they were sleeping, and they found that the traces that were recorded presaged the more complex renditions they were going to give the next day or in the future. So it was as if in sleep the rehearsal process was making the song more complex with markers of signatures of what was going to come. So, I mean, fascinating.
It's absolutely fascinating.
But broadly, it's a very complicated way of saying I don't really know, and nobody.
Somebody asks, how can I train my brain to be more sensitive?
So anything improves with practice.
Right.
So a simple definition of sensitivity is being able to discriminate, say, rough surfaces and smooth. When you practice at something, you become better and better at it. It's that, right? When you, people who look at, color swatches, cloths, and so on, are more sensitive to the small differences between them. People who smell perfumes or taste wine. So the experience of training makes you better at something.
Right. And two very quick questions, because we've got 2 minutes to the end of session. So till what age can the brain be trained?
Forever. Forever, but it gets harder. Young brains learn faster. We all know that.
Right.
At the age of 50, I began learning keyboard, and oh my God, how hard it is to learn to read music and translate it. I enjoy the struggle. I can feel my neurons, So you can learn forever, and the more you try, the better you get at it. But yeah, I wish I were a baby again.
And how do emotions connect to our sensory circuitry? That's the last question.
Emotions very strongly impact how you remember something. So if there is a strong emotional experience associated with something, you're going to remember it in a richer detail, more intensely, and then remembering every single thing that happened out there that didn't have emotional relevance to you.
Right. Wow. That was as if I've just had like 10 shots of espresso to wake up my my sleep-induced stupor this morning. It's been fantastic. Thank you so much, Dr. Tole. I'm actually out of words. So I'm just quickly going to move into my pre-prepared text. So huge thank you. It was really fascinating. Our next session here in 15 minutes is The Writing of Why Men Rape, an Indian undercover investigation as a feminist act by Tara Kaushal. And later tonight at 6 p.m. is our famous debate. This year's topic is Indian democracy is in danger. So remember to join us for that at 6 p.m. It's been chaired by Veer Sanghvi. You can buy the books that we speak about at all our sessions, in fact, from our bookstore partner, Tata Click. Our festival social media handle across all our platforms is @Tata Lit Live. Huge thank you to our title sponsor, Tata, as well as our session sponsor, the Blank Slate Knowledge Foundation, Mumbai. A huge thank you for being here as part of the 360 at Campus Festival, which is part of Tata Literature Live 2020. You can log on to tatalitlive.in, mark out all your favorite sessions and make sure you don't miss any all the way through till Sunday, because we have a jam-packed schedule and we have a stellar lineup of speakers and authors and ideologues from all over the world. That's the great part about going digital this year. So stay tuned and stay safe. Thank you so much for joining us this morning.